My name's Dr. Kesley Attridge. I'm a lecturer in the School of Biomedical Sciences and Physiology here at the University of Wolverhampton, and the project that we're looking to establish here is looking to investigate the interactions that occur between CD4 T cells of the immune system and cancer cells, so that we can try to identify how cancer cells are able to shut down the immune response, and hopefully we can unlock the potential of the immune system then, so that it can respond and fight back against cancers. My background is in the immune system, in immunology. So I'm a lecturer in biomedical science here, but my focus is very much on immunology. So I've always worked with CD4 T cells, which are a specific cell type of the immune system, and those are usually involved in fighting off infection and that sort of thing. But they're also, we know, involved in things like autoimmune diseases, where your immune system goes wrong or goes awry in some way, and you start attacking your own tissues, things like type 1 diabetes. So that's my background, CD4 T cell immunology. In the field recently, there's been a lot of interest in how CD4 T cells interact with tumours and with cancers. And there's been a lot of success in looking at the pathways and the mechanisms used by cancer cells to inhibit T cell responses, inhibit CD4 T cell responses, because what normally happens, or what we would hope to happen, is that your immune system would recognise a cancer as being a problem and would attack it, would try to fight it off and to kill it. But what we also know is that cancers have developed mechanisms to stop those CD4 T cells from responding to them. Either they mask themselves so that they aren't detected by the T cells, or when a T cell recognises them and tries to kill them off, the cancer cell produces factors, things called cytokines or small proteins, that prevent them from functioning, prevent the CD4 T cells from functioning properly. And that's quite a big area of research at the moment. And here at Wolverhampton we have quite a big strength in cancer research. So the project came out of my interest in CD4 T cells, but also trying to take advantage of the kind of excellent research base we have here in cancer research, and particularly in things like brain tumour research. So what we're really trying to investigate is twofold. The first is we're trying to look at individual mechanisms used by those cancer cells to suppress the T cell response. And then by looking in different types of cancer, we're trying to see whether, if and when we do identify a novel pathway, whether that pathway is used by just that particular type of cancer, or whether it's shared by lots of different types of cancer. Because in terms of therapies and stuff for cancer, if those pathways are used broadly by lots of different types of cancers, then that will make any therapy that's developed as a result of this more effective, in that it will be more applicable broadly across cancer types. Once we've identified these novel pathways, what we would be looking to do then in future is to develop things like antibodies that can come in and target those soluble factors made by the cancer cells that will inhibit the T cell response, so that the T cell never actually sees these factors that are secreted, and their responses are never shut down. They are freely able to respond as you would want them to, and are able to fight off the cancers. If we can develop those antibodies and show that they're effective, the aim would be to get those antibodies into clinical trials, and then ultimately to translate that science into a real benefit for patients by administering those antibodies to people suffering from cancers, so that we can hopefully help treat them and help make them better. So I think there would be an immediate benefit in terms of, yes, we've identified a novel pathway here, utilised by cancer, and once that data is published, that's open for other researchers to utilise as well, and in terms of informing our thinking about the ways in which cancer cells interact with T cells, so that in the future we can develop better cancer therapeutics. Because at the moment we still are stuck in an age where we're targeting cancer in a very non-specific way, and it remains a big problem that when we use things like chemotherapies and radiotherapy, we do a lot of damage to surrounding healthy tissues. And the idea with developing, in understanding these sorts of pathways and these sorts of interactions that this project aims to look at, is so that we can make more targeted therapies that are A, more effective, but B, are less damaging to healthy tissue. So being treated for cancer isn't such a horrible experience. I mean, what I really want to do is follow this through for the long term. So once we do identify a mechanism, not just see whether it is shared by those cancer types, because any new mechanism, even in just a single cancer alone, that mediates suppression of the immune system, must be a really important target for us to look at. So, following on from this, I really would like to try and see, antibodies to those pathways developed, and then you're getting into the territory of things like clinical trials and so on and so forth. Ultimately, hopefully, this is the aim of translational medicine, is to do this basic research and translate it so that it can be utilised in the clinic, and that's what, in the longer term, that would be the major aim for the project for me.